Hey everybody, welcome back to another Glass Bio video. This week we're going to have a couple of videos. This is the first of two mutations videos. The second will deal with larger chromosome mutations. And we're going to talk about today are mutations. And a mutation in general is just a change in the genetic material of a cell. And more specifically today, we are going to talk about point mutations. And a point mutation is just a chemical change in one base pair of a gene. So we're looking at a mutation on the gene level. So when we talk about point mutations, we're talking about two basic classes of mutations. The first of which being substitutions, and the second of which being indels, which are composed of insertion and deletion mutations, respectively. So the first type of point mutation that we're going to take a look at will be the substitution mutations. And substitution mutations occur when a single base pair in the DNA has been substituted for by another base pair. So the image that we see here is a sequence of nucleotides, and the only sequence that you're seeing is the antisense strand. So the sense strand is not shown because in this case it's not very important that we see that. So this sequence on top is the normal sequence, and notice that the fourth triplet here is highlighted in red, and the sequence is supposed to read GAG. Now if we look at the mutated sequence on the bottom, this sequence reads GTG. So the only difference between this strand up here, which is the normal strand, and this strand down here, which is the mutated strand, is the substitution of this T for that A. And what we see here is that because we have a substitution, we end up with a different amino acid. And this difference is enough in this particular case to cause sickle cell anemia. So the second type of point mutations that we talked about in class were called the indels. And the indels include the insertion and deletion mutations. And insertion and deletion mutations are exactly what they say they are. An insertion mutation is when you insert a base pair into a gene, and a deletion is when you delete a base pair from that gene. And both of these will result in what are called frame shift mutations because they disrupt the reading frame of the codons that we're going to use to create our proteins. And this will cause all the codons from the point of the mutation to the end of the gene to code for different amino acids. And generally what you're going to get as a result of this is a non-functioning protein. So let's take an example. What we have here is a sequence of nucleotides. So the blue on top represents a strand of DNA or gene of DNA. So we just have a randomly put together sequence of DNA. And underneath that here in red, we have the mRNA that would be transcribed from that. So generally what you would see is if we were going to translate this, it's going to read these in groups of 3. So that would be the first codon, this would be the second codon, this would be the third codon, that would be the fourth codon, etc., etc. And if we actually did this, that AAC is going to code for asparagine, as you see we have below. GUA is going to code for valine. The CCA is going to code for proline. And this is how you would translate this. And this is what I mean by the reading frames. Each codon here is a reading frame. So tyrosine, and then finally we're going to put together, or come down and get our aspartic acid. So this would be what we would normally get if we had this gene and this gene wasn't mutated. But let's say now that we come in here and we take this particular GC and eliminate it. So if this GC, and let's ungroup all this, if that GC were eliminated, get rid of that. If this GC now were gone, right, we've deleted that, and by deleting that, we're also going to delete, and again, let's ungroup all this. We're going to delete this G. Right, so that deletion right there is now going to change how we read this. So we have to relook at how this sequence is going to be read. So put that back. Our AAC is going to be the same, so that doesn't change. But now here's where our frame shift comes in. We're not reading that frame anymore because that piece of it is gone. So now we have UAC, which codes for tyrosine. We have our CAC, which codes for our histidine. We have our GAU, which codes for our aspartic acid, and our ACG, which now codes for our 3 and 9. So if you look back, this sequence of amino acids is different than the original sequence of amino acids. And this would go on until we reach the stop codon or until we got to the end of our sequence of mRNA. So that's a frame shift. We're just changing the frame that you're seeing that's being read because of an insertion or deletion mutation. So the point mutation that we've just discussed will have varying effects on the proteins depending on where they occur and if and how they change the resultant protein. Substitutions and indels may result in either missense, nonsense, or silent mutations. So the first one that we'll look at are missense mutations. And a missense mutation results in the substitution of one amino acid for another. And if we look back at our insertion image that we have here, we can see a clear example of a missense mutation. So this is an example of an insertion mutation, and all we've done in this insertion mutation is inserted an A here, and by inserting the A into this sequence of DNA, we've changed this particular amino acid. So we've gone from this to this, which is now going to change every one of the amino acids from this location down to the end of this particular sequence of amino acids. Now, having said that, not all missense mutations can lead to major protein changes. If an amino acid is replaced by another amino acid that has similar properties, the resultant protein will likely remain the same. And I'm not going to ask that you know which amino acids have similar properties. That's just too much to know. And finally, missense mutations are responsible for a vast majority of the mutations in the P53 gene that we talked about when we discussed cancer. So a full 30 to 50% of all cancers occur because of some type of missense mutation. So the second result would be a nonsense mutation. And a nonsense mutation is a mutation that results in the coding of a premature or early stop codon. And the resultant protein is not as long as it should be and is usually non-functional. And what we see here is a clear and obvious example of that, because we're supposed to have this long sequence of amino acids here, but because of a simple substitution here, what we're going to get is a early stop codon. So the protein here is obviously much shorter than this protein, and that shorter protein will probably be or will be non-functional. And the last set of mutations that we'll talk about are the ones that have the least effect, and they're called silent mutations. So a silent mutation is a mutation that does not change the sequence of the amino acids. So they may occur in the introns, they may occur in the exons, and here's an example of one that might occur in an exon where we've changed this GAA on the DNA strand to GAG, and eventually GAA and GAG code for the exact same amino acid. So this is not going to have any effect on the protein that is produced. However, new research has suggested that these silent mutations may have some kind of an effect on RNA splicing or the control of transcription, so the term silent mutation may in fact be a misnomer.